Hi, welcome to IB Chemistry SL Option D, Drugs and Medicines, video number 5. Today we're going to be talking about antibacterials and antivirals. So we're going to start with antibacterials. As the name indicates, they are drugs that kill bacteria. But before we talk about them, we need to tell you a little bit about bacteria. Bacteria are single-cell organisms that can be aerobic or anaerobic, i.e. they can live in the presence of oxygen or in the absence of it. And they generally have this particular structure in which they have a cell wall, which is external and is composed of sugars, lipids, and peptides. You have a plasma membrane, which is actually composed of phospholipids, and that's really what most closely envelops the cell. The cytoplasm is the liquid part of the cell, and it's the cellular solution where all the reactions and processes are taking place. And it contains one circular DNA molecule as a form of genetic material. So it basically means that it has one chromosome, very different from that of eukaryotic cells like the ones make us, that have many more chromosomes, plus they are not circular. In addition, you will find floating in the solution ribosomes, which are the sites in which protein synthesis is carried out. All right, so here's just an image of what a bacteria may look like. We also have other little parts like the capsule that allows it to attach to surfaces, and we can have pili, and some of them will have flagella. But this is just a general image of what a bacteria looks like. Now, all bacteria are not necessarily pathogenic, meaning that they cause disease. Most of the bacteria that we have that are there are actually not particularly terrible to us, as long as we keep them outside of our body or we keep them in the right place. But we are concerned of pathogenic bacteria that actually cause disease. Now, we're not going to spend a lot of time on the history of antibacterials. Those of you who are in my class can read a little bit about that. But we're going to go directly to the discovery of penicillin and all the breakthroughs that came about afterwards. All right. So here's the thing. Penicillin was discovered in 1928 by Alexander Fleming as by mistake. All right. He actually had gotten some of his petri dishes contaminated with a fungus, in this particular case, the Penicillium notatum. All right, and he found that wherever he had that, he was trying to grow the Streptococcus aureus bacteria, and wherever the fungus was present, there was no bacteria. So something in the fungus, in the Penicillium notatum, had killed, all right, or had caused the prevention of bacteria growing and reproducing. All right, so actually kills the bacteria. So as Fleming goes on to figure this out, he realizes that the penicillium must produce something that kills bacteria. Fleming's work was continued by two medical professionals, Howard Florey and Ernst Chain. Basically, they took the compounds that were being made by the Penicillium notatum, and they actually realized that it wasn't the living Penicillium notatum that caused this, but actually something that it produced. And they were able to isolate that the pure compound there was the penicillin compound. All right. So Chain was instrumental in the isolation and purification of the chemical, while Florey did actually the human trials and actually saw that it acted by killing bacteria directly. So these are the two other people that with Fleming were awarded the Nobel Prize for their discovery of penicillin. In 1945, Dorothy Hodgkins actually was able to determine the structure of penicillin G. Penicillin G is one of the original penicillin that was discovered, and based on that, we were able to then go ahead and modify penicillin to be able to make more versions that would remain active. All right. Now, how does penicillin generally work? And here's just a general structure of penicillin. R is just the different alkyl groups that we're going to have. So we'll talk more about this. But penicillin basically works this way. All right. And I'll come back to the image above in just a moment. Penicillin has a core that is called the beta-lactam ring. All right, we can see the beta-lactam ring over here as well. The beta-lactam ring is actually a four-member ring, a heterocycle ring that is very, very stressed. All right. Now, notice the shape of this particular compound. All right, the penicillin. One part tends to mimic one amino acid, cysteine, while the other part tends to mimic another amino acid, valine. So they tend to mimic the structure of a dipeptide. Two amino acids bonded together have what we call a dipeptide. Now let's look at some of the functional groups that are present here. All right. Let me go ahead and circle them actually in red. This particular group happens to be an amide group. All right, we have one amide group there and a second amide group over here. The sulfur is causing what is called a thioether. And in addition, we have a carboxylic acid. So the penicillin core has all these different important groups in addition to having those heterocycles. Now, the way that penicillin is going to work is that if I can bring you back to thinking about bacterial reproduction. When the bacteria reproduces, it will replicate its DNA, it will form all of this. But one of the things that it needs to do, it needs to form a new cell wall between the two daughter cells, and this is where penicillin is going to act. So notice here that the beta-lactam ring is very highly stressed. It has 90-degree angles when the angles there would prefer to be 120 or 109.5. Remember this from hybridization, from the angles when we talked about geometry. All right. So what happens is, in the presence of penicillin, the bacteria normally use an enzyme called transpeptidase to actually work and building the cell walls and actually making bonds between the peptides in order to keep the structure of the cell wall, waterproof. All right, but because the structure of penicillin so much resembles some of the amino acids, penicillin will react with the transpeptidase. The beta-lactam ring, which is very stressed, will open up and bind to the enzyme, inactivating the enzyme. So what happens is that the bacteria no longer can form cell walls, and when the bacteria cannot form the cell wall, water flows in. It causes lysis because the cell membrane will actually burst. All right. Now, that's basically the general way in which penicillin will work. Now, if we misuse penicillin, because it has been over prescribed, we don't finish our cycles of antibacterials because we give it to animals and all these kind of things. There has been a growth of bacterial resistance. So bacteria have developed enzymes that are called penicillinases, all right? And basically what they do is they have an enzyme that will bind to the beta-lactam ring before it has a chance to bond, the penicillin has a chance to bond with the transpeptidase. And so it does not interfere with the formation of cell walls. All right. So how does it happen? Well, there's always natural resistance that is going to happen. Random mutation can cause the formation of this penicillinases, all right, that will actually cause the breaking of the beta-lactam in penicillin, all right. There's also the fact that if we don't kill all the bacteria that respond, all right, we're always going to be leaving behind some of the resistant strains, all right. How can we fight resistance? Well, we change the R group, all right. Here are some of the options. If we have the beta-lactam, all right, we can change the different structures, and you can see we have the same general penicillin structure, but we have changed the R group, and that gives it, sometimes it will not bind to the penicillinase in the bacteria and therefore remain active. Obviously, this is not perfect, all right? Some of the other problems with penicillin, in addition that many bacteria have become resistant to it, is also that it causes allergies, all right? So ways to decrease resistance, all right, is to have a much stricter control on antibiotic or antibacterial prescriptions, all right, not to use it on animals or other things. Required patients to complete the course of antibiotics in order to reduce the number of bacteria that will survive, and that can and the fact that we are going to be keeping only the resistant strains. But the reality is that as we use this, it will continue to form resistance. So what we need to do is always keep ahead of the bacteria. We have to start making more antibacterial compounds.